So again, this is Bio 212. We are looking at Chapter 27, Electrolytes and Acid-Base Balance inside the human body. And you're at Norwalk Community College. And we have thus far gotten through how do we actually measure the concentration of body fluids as a result of regulating water intake and excretion. Okay? And what we're going to do is we're going to spend a couple minutes looking at the intracellular fluid versus extracellular fluid and take a look at what's going to happen as we change the volumes of fluid using different hormones inside the body. So let's move from there. Let's go to here. Woohoo! There we go. So extracellular fluid is most abundant in sodium ions, and the anion is, of course, chloride. And to think about this, remember this was a cell. Let's go back to AMP1. Let's just draw a cell for you. Okay, cell. AMP1, prototypic cell. You're never going to see it in biology. Yada, 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 blah, blah, blah. Right? Okay, there's that. I'm going to go like this. Go like this. Have some vesicle. Where this is the nucleus. This is the rough endoplasmic reticulum. I'm going to put little ribosomes inside here. Right? Whoops. Here we go again. Don't freak out. Capture cursor. Nope, don't want to do that. Draw and zoom. Anyway, so here's our cell. These are going to be a rough endoplasmic reticulum here. That's the Golgi apparatus. And remember, in AMP1, we said there's sodium potassium ATPases, and their job is to have Na outside here. Okay, inside here. And we're going to have some chloride kicking around outside here, right? Okay, so this is what your textbook is trying to make sure you understand. You also now, because we're inside Chapter 27, you're going to notice I'm going to start putting this inside here. We haven't spoken about this buffering system, okay? But you'll see why later when we actually need to think about it, okay? So having said that, okay, I just put a plus on that. It's supposed to be minus. Sorry about that. The sodium-potassium pumps play a major role in keeping potassium ions high inside the cell and sodium high outside the cell. Okay, remember this is actually how we get our resting membrane potential on cells. When we say that, what we also want to make sure you understand is the consequence of this when you look at all of the different ions and protein anion concentrations, you can now see that if the color code here is telling you something, the extracellular fluids are the blood plasma and the interstitial fluid, as well as intracellular fluid. So let me just draw a cell for you again. Cell. Cell. All right, there's that. And here's that nucleus again. That means yellow. Let's go close to the color they have inside here. Inside here. That's the intracellular fluid. Whoops, not outside. Jonathan didn't get out of kindergarten. I was that kid who couldn't draw inside the lines. Anyway, and that means outside here in red is going to be material that's coming out of a blood vessel, okay, and traveling into a space and or being stuck inside the blood vessel. And then in blue, closest to the actual cell itself would be the interstitial fluid. Okay, around it right here. Yay. Okay, and as you move there, you're going to have that membrane, and that's where we actually change the ion concentration. But you can see pretty much, you follow this along, you're going to learn that potassium is the major ion intracellularly, sodium and chloride, major sodium chloride, major ions outside of the cell. So this should actually make sense. Okay, cool. Let's get rid of the cartoon. Let's get on to the next. What we're going to be doing here is I'm just going to point out to you primarily how we control sodium movement, right? So it's the most abundant extracellular fluid. It's controlled by aldosterone, ADH, and atrial natriuretic peptide. Okay? So we've seen these so many times this semester that maybe you're a little numb to it, but the reality is this is how we control sodium. And wherever sodium goes, guess what? Chloride goes. Okay? So it's the most prevalent anion inside the extracellular fluid. It helps balance levels of anions in different fluids. Okay, bold. Think of the chloride shifts inside of red blood cells, right? And antidiuretic hormone, okay, governs the extent with which water loss occurs from urine. So in this case, the process that increases or decreases renal reabsorption also affects the reabsorption of chloride. Because remember, just like I said a moment ago, wherever sodium goes, chloride follows it. Potassium, okay, is the most abundant cation in the intracellular fluid. Okay, it helps regulate body pH, okay, when exchanged for hydrogen protons inside the kidney. So let's go like that. Kidney. Okay. Don't know why it came on as a that right there. Let's get out of there. Don't know what the hell's going on. Okay, so let's go like that. Let's go like that. Okay. And notice something different here. It's controlled by aldosterone, and it stimulates the principal cells inside the renal collecting ducts. Okay. And way back when, inside AMP1, if you took the class with me, I told you, well, we really have to do is we have to regulate excess potassium ions inside of the body. And the significance here, of course, is if we don't regulate those, we can't maintain the conductance at a cell membrane. If you can't maintain conductance at a cell membrane, then you lose not only functioning, say, of your nervous system, which is paralyzing in and of itself, but you know fine well what will happen to cardiac muscle function. Okay? Meaning it will stop. We start getting to bicarbonate, and everybody remembers this. Okay? I could just go like this. Let's go H2O. All right, H2O plus CO2 in the presence of carbonic anhydrase is going to give us first the acid H2CO3, right? And that spontaneously dissociates into H plus HCO3, all right? And now you are reminded once again where bicarbonate comes from, okay? It comes from excess carbon dioxide. So it's the second most prevalent extracellular anion, and what it's going to do, of course, is picking up carbon dioxide. We form that carbonic acid. Carbonic acid, so I don't want to do that yet. Carbonic acid, I'm going to underline that for you. Where, remember, it travels back to the lungs where you're going to exhale that carbon dioxide. But remember, what's going to happen here is the chloride shift maintains the correct balance of ions between extracellular fluid and the intracellular fluid, and this is why chloride moves with carbon dioxide. Okay? If I teach you something new here, it's going to be that the kidneys are what's going to be responsible for regulating bicarbonate, and that's really important. We're thinking about metabolic versus respiratory compensation. Okay, and this means that if the kidneys are failing, it means we can't regulate bicarb. Okay, that's the real important idea here. Okay, so I want to make sure you understand that and you hear that, because inside the tubules, inside chapter 26, you saw how we collected bicarb when we needed it, or we could excrete it if we didn't need that much of it. Okay. Let's get rid of that. Calcium, our favorite friend from AMP1, not so much AMP2 unless we're thinking about cardiac muscle function, okay, is regulated by parathyroid hormone. This is a great opportunity for us to remind ourselves that the parathyroid glands are posterior to the thyroid glands. They detect the amount of calcium inside the bloodstream, and when low, it increases parathyroid hormone, and that's going to stimulate osteoclasts to break down bone by resorption. Okay, it also enhances resorption from glomerular filtrate. Okay, so that means we pick up more calcium, we make sure we don't urinate it out, but the part that most people forget is that it also increases calcitriol production. Okay, something just happened there. Whoa, spinning wheel of death. Okay, not what I wanted to have happen, but I'll take it. Okay, we'll take that too. Okay, so parathyroid hormone targets kidney, right? It also targets osteoclasts, And calcitonin acts antagonistically to that. So calcitonin will trap that calcium by activating osteoblasts and shutting off osteoclasts. Phosphate is the new member, and don't want to spend too much time thinking about it, but just remember that if you're regulating calcium, you're regulating it by using parathyroid hormone, calcitriol, and calcitonin. Okay, underline. Okay, and don't forget about calcitonin. I'm just going to put this in brackets over here. Right, because it would be antagonistic. Okay. Magnesium. We don't think about magnesium enough inside this class, but it's a cofactor for certain enzymes, and most importantly for the sodium-potassium pump back in AMP1. It's essential for normal neuromuscular activity, synaptic transmission, and myocardial function, and the secretion of parathyroid hormone depends. Did you see that? Depends on magnesium inside your bloodstream. So if you're not making enough parathyroid hormone, check magnesium levels. Okay. What we're going to do now is we're going to switch, and we have all these ions kicking around inside the body. What we need to do is now, in the last few minutes of all of this, we need to think about what it means to have a pH range. And I know everybody knows this right here. All I have to do is go like this, go like that, and everybody goes, oh, he's talking about homeostasis again. Right, well, remember, that pH range is so we maintain the correct 3D shape of proteins. And when you don't do that, well, guess what? Those proteins don't have the right shape, and therefore they can't do their job, which is keeping you alive. Okay? So what do? We have those buffer systems. They act to quickly, temporarily bind hydrogen protons. They can raise pH but do not remove the protons so that we can actually utilize them some other place. And what we have are protein buffering systems. And you think about that. You actually have hemoglobin. Think about what hemoglobin was actually doing back in chapter, what was it, 19, right? It was binding hydrogen protons. And then again in chapter 23 in respiration, it was the shift in the pH that would allow us to release oxygen from that hemoglobin. Likewise, albumin can act as a buffer. And what's happening are the free carboxyl groups act like an acid when they're releasing hydrogen protons. The amino groups of all the proteins act as a base. Okay. Carbonic acid bicarbonate buffering system. How many times have we actually gone over this semester? I think maybe as many as the RAA pathway. The one that we haven't spent any time thinking about has been that phosphate buffering system. But really what I want to make sure you understand is it's an important regulator of pH inside the cytosol of your cell. So how do we get rid of things? Well, everybody remembers that to get rid of carbon dioxide means we actually lower the likelihood of making hydrogen protons. But what I want to make sure you understand is that carbon dioxide is called a volatile acid, and if we change anything in our breathing pattern, we change the presence of that volatile acid, and therefore we change the actual amount of hydrogen protons. So this is a really bad figure to look at. I'm sorry I can't make it any bigger than this, but inside your textbook, remember the blood pH is either increasing or decreasing, but inside this figure, it increases hydrogen proton concentration. Chemoreceptors detect it inside the medulla and the aortic as well as carotid bodies. And then we change the amount and forcefulness of carbon dioxide being removed, and therefore less carbonic acid forms, therefore less hydrogen protons, and therefore the pH will decrease. Okay. Imagine I'm at the board and I'm drawing all of those little arrows going up and down. The other way, of course, is to use the kidneys, where we can excrete. I'm going to bold that for you. Whoop, that didn't work out right. Okay, well, whatever. Okay, which is going to be metabolic reactions are going to produce non-volatile acids, and in this case we can get rid of that hydrogen proton in the proximal convoluted tubules. Got to hit pause here, the furnace just came on. And what you can see, of course, is that if we're going to use the kidney to excrete those hydrogen protons, we have to gate you to a specific location. The first is going to be the proximal convoluted tubule. We'll have a sodium hydrogen antiporter. Okay, so we retain the sodium. We excrete or secrete the, what just happened there? Let's undo that. Okay, go like that. We're going to secrete hydrogen protons. Those hydrogen protons Okay. We'll go to the collecting duct, okay, which actually will then also secrete hydrogen protons into the tubule fluid. So your urine can be 1,000 times more acidic than your blood, and the two buffers that are combining with it inside the collecting duct are ammonia, okay, and the phosphoric acid, nudge, nudge, hint, hint, wink, wink, okay. And you can see that here, where you can bind hydrogen, make urea, and over here we can pick up bicarb, and chloride, a bicarb chloride antiporter allows us to use carbon dioxide to push hydrogen protons out. Okay, so we collect the bicarb to buffer our bloodstream, remove the hydrogen protons into the space. Okay, and now we're going to buffer that. Okay, so if this is the intercalated cell, the collecting duct, you can see you actually have that mechanism here. Okay. What you're doing here, of course, is buffering those ions there. So, what's the consequence of this? Because that's really what you want to be thinking about. Here's you, the end of all of this, 2 semesters of anatomy and physiology, you are depleted. You've had so much work to do, you haven't been allowed to drink water, and you're going to push yourself out of normal homeostasis, right? Okay, or too much running around and overexcitability, so you're either going to depress the synaptic transmission if you enter acidosis, or you're going to reach overexcitability of the central nervous system as you become alkalytic. Okay, and that's because your pH ranges have shifted outside of that. And so right now, let's go like this. It's you like that, and you're like bumming. Okay, fantastic. What can we do to fix that? Well, let's go like this. Get rid of that. Go here. You can normalize arterial blood pH. You can have a complete restoration, usually by compensating. Okay, and this would be just normal. Okay, this would be return to normal. Return to normal. Norm. Parentheses there. Parentheses here. This is where your textbook gets a little bit weird, right? It says there can be a partial compensation. And that, what they mean there is if you got values of a patient who had, say, a carbon dioxide value, pH value, and say bicarb, HCO3 minus value, that were outside of their normal range, but the body was trying to get back to normal, and you still had one of these values either still too low or still too high. And that's what they mean by partial compensation. Okay, so your kidneys are failing. Okay, so your lungs are going to try to increase the amount of carbon dioxide being excreted. The pH may go back to its normal range, but your bicarb may not be in its correct range. That would be partial compensation. Respiratory compensation, it would be either hyperventilation or hypoventilation. And renal compensation is going to be secretion of hydrogen protons. Notice I'm saying secrete, so you pee out more hydrogen and you reabsorb more bicarb. So if the renal system is compensating for a failing set of lungs, you'd expect to see a higher concentration of bicarb inside the body if at all possible. Okay, so let's see. Okay, so respiratory acidosis, abnormally high partial pressure of carbon dioxide in systemic arterial blood, and that's because you're not exhaling enough. Okay, you're either holding your breath, you're taking an exam, and you're freaking out, right? Okay, you could have emphysema, airway obstruction. The kidneys help raise blood pH here, okay? And what's going to happen, of course, the goal is going to increase the exhalation through ventilation therapy. Okay, if you can't get the obstruction out of the way, nothing good is going to come of this. Respiratory alkalosis, low PCO2 in systemic arterial blood. Renal compensation, of course, can help here because you can actually get rid of some of the hydrogen protons, okay, increase the number of bicarbonate ions that are retained. But a simpler treatment, of course, is to simply breathe into a paper bag, okay? Because what's going to happen is if you have an abnormally low range for carbon dioxide, which was 22 to 26 millimeters of mercury, right? Okay. It's going to change the entire system. So if you increase the total amount of those ions, okay, in this case, the volatile gas, okay, you can actually now increase the amount of hydrogen protons. So the results from changes in bicarb would be metabolic acidosis, where you have an abnormally low bicarb value, okay, and the accumulation of acid other than the carbonic acid is going to lead to ketosis and failure of the kidneys to excrete that, okay, hydrogen proton, okay, is going to cause major problems. But the reality is going to be this, okay, hyperventilation can help, meaning blow as much carbon dioxide off, but the easiest thing to do, of course, not that any of us actually have these at our disposal on any given day, administration of IV, not IV sodium bicarb, okay? And at the end, I think this is our last slide, right? And yeah, oops, we're going a little bit over here. Metabolic alkalosis, okay, where you have abnormally high bicarb. Okay, you have a non-respiratory loss of acid, such as overdosing, vomiting stomach contents out, excess taking of alkaline drugs, antacids, severe dehydration, where hypoventilation can help, so hold your breath to increase the concentration of bicarb. And obviously, you could give fluids to correct chloride, potassium, and other electrolytes. But I want you to think about, you know, what it means to actually see. Let's go like this. What does it mean if you see somebody whose values, well, I'm going to just put it as a question. Do you know the correct ranges of each of the, I'm going to put this like this, CO2. And we're going to have HCO3 minus and pH. I know everybody knows the values of pH. Okay, that's a good place to stop. We're going a couple minutes over. That's okay. I hope this is helpful in understanding what chapter 27 was about. And if I have time, I'm going to start some chapter 18. Okay, have a great night. Take care. Bye.